All right, welcome back to the deep dive. Today we're taking a look at something that's super important in the world of microbiology. It's how scientists figure out if medicines can actually beat those nasty microbes that make us sick. We're talking about antimicrobial susceptibility
You
tests,
got it, or ASTs for short.
right, ASTs. And essentially, it's kind of like, trying to find the right key
Yeah,
to unlock a lock. But in this case, the lock is a bacterial infection.
that's a great analogy. At the heart of it, we want to see if a drug can stop or even kill a specific microbe, like literally in a lab setting. We take a carefully measured sample of bacteria and we expose it to different amounts of a drug.
Yeah.
Then we just watch what happens to their growth.
So you can see if it works or not.
Exactly.
So our mission today is to unpack all these different ways scientists do these tests.
Yeah,
Because there's a bunch.
here are.
And we want to give you a good understanding of how we know for sure if a drug is actually effective. So we'll be covering some pretty cool stuff. We've got broth dilution, agar dilution, then there's disk diffusion.
Also known as the Kirby-Bauer test.
Oh, right, the Kirby-Bauer test.
Yeah.
Then we've got time-kill kinetics, and then something called bioautography.
All right, let's dive right in. We can start with broth dilution. Now, the main thing here is figuring out something called the MIC.
MIC.
Yeah, MIC. That stands for minimum inhibitory concentration.
Oh, okay. So what that means is it's like the smallest amount of a drug you need to stop the microbes from growing, like visually growing, in a liquid broth.
You got it. You take the antimicrobial and you make a bunch of different concentrations, each one weaker than the last. And these are in a liquid growth medium, kind of like a soup for the bacteria. Then we add a specific amount of bacteria to each of these concentrations. Usually it's about 500,000 CFU/ml.
CF.
Yeah, CFUs, those are colony-forming units per milliliter.
Okay, so basically a way to measure the number of bacteria.
A measure of the viable bacteria. So we let that sit overnight, and then we check for cloudiness. If it's clear, it means there's no growth, and the MIC is the lowest concentration where that happens.
Ah, so you can actually see with your own eyes where the drug is doing its job.
Exactly. And this method, you can scale it up or down.
Oh, really?
Yeah, you can do macro dilution using test tubes, or micro dilution with those little 96 well plates, which is great for testing tons of combinations, and you can even automate it.
Nice, efficient.
Right. Another big plus is that it gives us a precise numerical MIC value. Plus, you can even figure out the minimum bactericidal concentration, or MBC. It's the lowest dose that actually kills the bacteria.
Oh, wow.
You just take samples from those clear wells, put them on fresh plates, and see if anything grows.
I see. Clever.
It's considered the gold standard for good reason, although you
Okay.
got to be really precise with those dilutions.
Yeah, bad.
And it might not work perfectly for every single type of microbe.
Now, agar dilution sounds pretty similar, but instead of liquid, you use something solid.
You're right, we use agar plates. Instead of the liquid broth, we mix the antimicrobial right into these agar plates. Each plate has a different concentration. Then we take our bacteria samples and put little spots of them on each plate.
Okay.
And the MIC is the lowest concentration on the plate where you see, well, no growth around that spot.
So basically, instead of looking for cloudy or clear liquid, you're looking for spots of bacteria or no bacteria on the plate.
Precisely.
I get it. And what's the advantage of this method?
Well, one big advantage is you can test a lot of different strains of bacteria all at the same time
Oh, that makes sense.
on one plate. And it's really easy to see if something is growing or not. The downside is that getting those plates ready with all those different concentrations, it's a bit more work than the broth dilution.
Right, makes sense.
Yeah.
Let's move on to the disk diffusion, or the Kirby-Bauer test.
Yeah, the Kirby-Bauer.
I feel like I've heard of this one. It's that one used a lot in hospitals and clinics, right?
Yeah, exactly. It's more of a qualitative test. You start by spreading bacteria all over an agar plate. We call it a bacterial lawn.
Like a nice even layer.
Yep. And then we place these little paper discs, each soaked with a different antibiotic, right onto that lawn.
Okay.
As the antibiotic spreads out from the disc, it makes this concentration gradient, kind of like when you drop ink in water. If the bacteria can't handle the antibiotic, you'll see a clear ring around the disc.
That's the bacteria dying off.
Right, that's called the zone of inhibition.
Gotcha. So the bigger the ring, the more effective the antibiotic.
That's the idea. We measure those rings, those zones, and compare them to, well, standardized charts to see if the bacteria are susceptible, intermediate, or straight up resistant to that antibiotic.
So it's like a quick and dirty way to see if a drug is going to work.
It's simple, doesn't cost a lot, and gives you answers fast. That's why it's so popular in clinical labs. But it's not perfect. We don't get an exact MIC number.
You just get a general idea.
Yep. Right. And things like, how fast the drug moves through the agar, it can mess with the size of that zone. So you got to keep that in mind.
Okay, so it's more of a screening tool.
Yeah, I'd say so.
Gotcha. Now, what about time kill kinetics? That sounds like it's looking at how the drug works over a period of time.
It is. Instead of just a one-time check, we monitor those bacteria as they're exposed to the drug. So we add the bacteria to a broth that has the drug in it. And then we take samples at different times, maybe every hour for a few hours, and then at 24 hours just to see what's happening.
Like a time lapse.
Exactly. We count how many bacteria are still alive in each sample. And when you plot those numbers over time, it shows you how fast and how effectively that drug is killing those bacteria.
So you can actually see if a drug is a quick killer or if it just stops them from growing.
You got it. That's how we tell the difference between bactericidal drugs, the ones that kill a lot of bacteria, and bacteriostatic drugs, which just prevent them from multiplying.
So it gives you a much more detailed picture.
It does, and it's great for research, but it takes a lot more work. All those samples and counts.
Yeah, a lot of lab work.
Yeah, and the analysis is a bit more complicated, too.
All right, and finally, we've got bioautography. Now, that sounds like a pretty fancy technique.
It's pretty cool. This method is used when you have a mixture of things, like, say, an extract from a plant, and you want to see if any of those things have antimicrobial activity.
So like finding the active ingredient in a natural remedy.
Exactly. So you start by separating all those components in the mixture. That's where chromatography comes in. Once you've got everything separated, you basically put it on a plate with agar that's got the test microbe in it. If any of those separated components can kill or stop the microbe, you'll see these clear zones right on the plate.
That's pretty awesome. So you can pinpoint exactly which part of that mixture is the potent one.
That's the idea. It's really useful for discovering new drugs from, natural sources. Of course, you need the right equipment, and the results are more qualitative. It tells you if something's active, but not exactly how strong it is.
That makes sense. So it's more like a first step to finding promising candidates.
Exactly.
Well, we've covered a lot of ground today. We've talked about all sorts of different methods for figuring out how well drugs work against microbes.
And each method has its own strengths and weaknesses.
It depends on what you're looking for.
Right. What specific information do you need? What resources do you have? And what are you actually working with? What drug, what microbe? All of that plays a role in choosing the right method.
This has been a really fascinating look into this world of microbial assays.
Absolutely.
And it really shows how much work goes into understanding how to fight off these infections.
And it makes you think, right? I mean, all these different approaches, from the simple ones where you just look and see to the more complex ones where you track things over time, they all contribute to the fight against these microbial diseases. And it's all part of the effort to find the next generation of antimicrobial agents.
It really is a constant battle.
It is.
But it's fascinating to see the science behind it. Well, that's it for our deep dive today. Thanks for joining us.
Thanks for having me.
And we'll see you next time.
See you then.